Good afternoon. We're going to get started here. So thankful that you guys can all join us. This is a little bit of a different session than some of our other ones. Normally when we have the conference, we get a lot of the sessions from the companies that are running clinical trials, and that's what the families are hearing about that, or some of the approved therapies. We wanted to take an opportunity to share with the community some of the research that PPMD funds, and in part that the families help us fundraise to support, so you can see kind of where your contributions are going and how we're helping to move the field forward. So it's a little bit of a different mix of presentations today, but we hope it's something maybe a little bit different and that you'll enjoy it. So our first speaker is going to be Matthew Lumley from Myosana Therapeutics. So this is actually part of PPMD's recent kind of venture philanthropy efforts. We provided some investment to Myosana. So I'm going to have Matthew come up and start us off.
Good afternoon. Can everyone hear me? Cool. So firstly, lovely to meet you all, and it's a real pleasure to be here, and a big thank you for PPMD for inviting me. So my name's Matthew Lumley, and I'm the CEO of Myosana Therapeutics. So really just over the next 10 or so minutes, it's really just a high-level introduction to Myosana, what we're doing, what our objectives are, and what our approach to DMD is. This isn't an in-depth scientific presentation. We'll touch on some elements, but it's very much an intro to us. So Myosana is a biotechnology company. We're preclinical, which means we're not performing clinical trials. We're developing a drug, which we hope to take into clinical trials in the future. And we are a spin-out from the University of Washington, but still the large majority of our activities happen within the University of Washington in Seattle. And the two founders are shown on the slide here, so Stan Frohner and Nick Whitehead. So Stan's a professor of biophysics and physiology at the University of Washington. He's worked in muscular dystrophies and DMD for over 30 years. Some really significant contributions to basic science. And in more recent years, he's done a lot more translational medicine and drug development. Nick Whitehead is a muscle physiologist from Australia, but has been in Seattle for 15 years and understands tutelage, is really the inventor of the Myosana platform, and I'll talk about the platform later in the presentation. And as mentioned, my name is Matthew Lumley. I'm the CEO, and my background is I'm a clinician scientist, so I've trained and practiced clinical medicine in London, adult internal medicine, general cardiology. I did my PhD at St Thomas's and King's College in London, and I've spent 10 years working in drug development. So I've been working in the rare disease space for the majority of the last 10 years, initially at Pfizer, then at Moderna, and now I split my time between Myosana, and I also work for a life sciences venture capital group in London, so investing in biotechnology companies. The back end of last year was a real sort of inflection point from Myosana in terms of a maturation of a company, and really transitioning from being an academic group with an exciting idea to becoming a small but a fully fledged company. So we closed our first significant funding round of just over $5 million. And the objective of that funding is really to select a drug candidate that we'd like to take forward into clinical trials. We've got a fabulous, really great set of investors. So our lead investor is a chap called John Ballantyne. He's been intimately involved with gene therapy for 30 years. Many people in the biotech world will know him, and he's been a real champion, and he's led the investment. But we've also had really long-standing support from PPMD in particular, who made a very early investment in Myosana, and also invested again in the recent seed round through their venture philanthropy. We've got funding from the Muscular Dystrophy Association, and we also had some early funding from Cure Duchenne Ventures. I wanted to touch on my personal connection to Myosana and DMD. So really, why is a physician from London the CEO of a drug company in Seattle with an 8-hour time difference? And, you know, at the crux of it is Miles, who's in the middle of the picture. So Miles is my son. And about 3 years ago, he was diagnosed with Duchenne muscular dystrophy. And I think all the parents in the room know what that means and the kind of the, you know, immediate impact that has, and it changes everything. And Miles is here with his sisters. And, you know, I love this photo. And again, I think this will resonate with many of the parents. These photos were done just after he started steroids. So we wanted to capture some really special pictures, you know, before he had the chronic impact of corticosteroids, change of appearance, and etc. So I think, you know, my purpose and connection should be fairly obvious to why I'm doing this. Now, you know, why Myosana? You know, our aim at Myosana is something transformational. You know, and as a parent, I've been on that journey. You know, what would the ideal therapeutic look like for DMD, and how might it work? And you know, AAV and microdystrophin is the here and now of gene therapy in Duchenne muscular dystrophy. I strongly believe it's not the future. And I, you know, as someone who knew the drug development space and knew AAV well and followed it for years professionally, but didn't have a personal interest in it, when it came to, you know, my son, I had concerns. You know, concerns about safety, concerns about not giving full-length dystrophin, the lack of ability to repeat dose, and, you know, the potential to closing doors of, you know, a single treatment. So, you know, the philosophy of Myosana is we'd like to, as best we can, overcome those key limitations of AAV-delivered microdystrophin. And there's two, you know, fundamental principles we believe that a transformational therapy for Duchenne muscular dystrophy would have. And first and foremost is that we believe that full-length dystrophin delivery is essential. Now, that I caveat that with, it's not to say that I don't think there's a role for truncated dystrophins. It's not that I don't think there's a role for microdystrophin. But we believe that the dystrophin is a far more complex protein than is often discussed. It's more than a mechanical protein. It's not simply a shock absorber or a carabiner. We see it as a scaffold off which many proteins anchor themselves. And all of those proteins that form this complex with dystrophin have critical functions in, you know, in maintenance of the cellular environment, signaling function involved with vasodilation, ion channel regulation, many, many more functions. So to really normalize that cellular environment and stop the progression of the pathophysiological disease, we think full-length dystrophin. Full-length dystrophin is essential. And there's much supportive evidence for that. It's really substantiated in the literature, from the preclinical literature, genetic cohort studies, and I think we're seeing glimpses of that from the magnitude of effects that we're seeing with microdystrophins. The other thing we think is critical for gene therapies that are not integrative, i.e. gene therapies that don't become encompassed in the host genome, is that repeat dosing is essential. You know, muscles grow, muscles divide, and, you know, a fundamental part of the pathophysiology of Duchenne muscular dystrophy is repeated injury, inflammation, and pathological regeneration. So as boys grow, as they have repeated injury, the impact of or the effect of a single dose gene therapy will be diluted. And thus, if you really want to maintain the benefits you have, you need to keep on topping up that dystrophin you're given. So we think repeat dosing is essential. So this slide shows a pictorial representation of our drug product. I'm not going to describe the components of it in detail, other than to say that it's a novel gene delivery platform. And our first target is Duchenne muscular dystrophy. But the principles of the Myosana therapeutic is it's non-viral, first and foremost. It's been designed to minimize immunogenicity and thus enable repeat dosing. We currently have an unlimited carrying capacity as far as we've tested it. So to date, we're able to deliver the full-length dystrophin gene intravenously. We've given repeat doses. We see our DNA within the myocytes, within the cardiomyocytes, we see protein translated, and we see that protein correctly located, and we also see functional benefits. So we're really excited about what we're seeing. The other neat element of the platform is it is highly specific to cardiac and skeletal muscle. So, that's again designed to minimize the doses required and decrease the off-target side effects that we're becoming very familiar with AAV. So I'm not going to go into the details of this slide, but really the crux of our intellectual property, really the genius idea that Nick came out of was how can we deliver our cargo specifically to muscle? This goes back to his background in muscle physiology, and the protein we've identified is called the GLUT4 protein. Now, this is something that most basic scientists will be familiar with. Medical students, physicians all know. The GLUT4 protein is involved with transporting glucose into myocytes and cardiomyocytes. So it's a naturally occurring transporter protein. Its whole life cycle of the protein is it sits on the transmembrane. It's insulin sensitive. It binds glucose. It forms an endosome. It comes into the cell. It releases glucose, and it goes back to the membrane. And that's its life cycle. So really what we're doing with Myosana is we're hitching a ride on that natural transporter mechanism. So we have an antibody that binds to an extracellular loop of the GLUT4 protein, and conjugated to our antibody, we have our DNA cargo and the other components of the drug product. So really that is the crux of the platform, at the highest level. So in terms of timelines and, being really cautious not to set expectations. The target we're internally driving towards is to select our first development candidate, hopefully early next year, but sometime next year. And really that's a real pivot point for the company, that once we have a development candidate, we can march towards the clinic. And with that, it enables us to scale up our manufacturing and do all of the requisite experiments that are required to go into man safely and confidently, so toxicology, pharmacology, etc. So here, we're really aiming to be in the clinic in 2026. I think there's an off chance that we could be in the clinic in 2025, but again, very cautiously trying to moderate expectations. So, thank you, and we take questions at the end. Cool, thank you.
Our next speaker is going to be Dr. Lee Sweeney from the University of Florida with the Florida Wellstone Center of Excellence. So this is something that for families, you may remember our holiday campaign of, I think it was 2 years ago, to provide some supplemental funding to UF.
Yeah, thanks, Erik. Yes, PPMD has provided support for our Wellstone beyond what our Wellstone Center was funded by NIH to do. And mostly that money's been used to try to further develop rodent models that better represent the human disease for the testing of potential therapeutics. So as I basically just said, that really the purpose is to be able to evaluate potential therapeutics in a model that looks much more like the human disease. And to that end, we've been characterizing both the severe mouse model of DMD, the so-called D2-MDX mouse, as well as a rat model of Duchenne, which has a very similar phenotype to the mice, but progresses much faster. And so the rat has advantages in that it's a larger animal, but disadvantages in that it's much more expensive. And also the rats turn out to be much more fragile than the mice. And so we've lost quite a few animals trying to do procedures that need anesthetics. So the two rodent models we have developed really exhibit key skeletal and cardiac features of the human disease, which is not the case with the more commonly used C57 black 10 background, so the common MDX mouse, which we were not happy with because of the fact that so many drugs that were showing efficacy in that mouse were failing once it got into humans. And so we thought largely the problem was the fact that the mouse model was just too mild. The skeletal muscle maintains very good regeneration until the animals get quite old. Low levels of fibrosis and TGF-beta compared to other backgrounds. And the cardiac disease is very mild until the animals actually have age-related cardiac dysfunction imposed upon the disease. So when they get old, they have more cardiac disease. So we figured that if we could develop much more severe models that look more like the human disease, or at least like severe forms of the human disease, we could show benefit of some treatment in that background, then it'd be much more likely to bring benefit once it got into the clinic. So this brings me to the point of the Duchenne cardiomyopathy. And one disadvantage of the humans compared to the rodents is that the humans show much more variability from person to person, probably because of other genetic modifiers of the disease, as well as the fact that the boys aren't all on the same drug treatments. And so the presentation in humans is not nearly as uniform as what we see in our rodent models, but we think our rodent models do recapitulate the more severe forms of the human cardiomyopathy. And people have characterized the fact that there is diastolic dysfunction that leads actually to a reduced end-diastolic volume, as well as one can see abnormal EKGs in some of the boys, as well as early infiltration of fibrosis, even before you can measure some of these other measures of diastolic dysfunction. And this diastolic dysfunction, in the absence of an ACE inhibitor, which actually does help prevent the fibrosis, does end up with a much smaller end-diastolic volume, and therefore and this is interesting because the ejection fraction is really not changed very much. Contractility is still pretty good. It's just now the end-diastolic volumes are down. But then that progresses to the point where you start to have systolic dysfunction. Ejection fraction starts to drop, and that's concurrent with dilation of the cardiac cavities, then the progression to out-and-out heart failure. And again, the ACE inhibitors do impact the timing of that progression, and they delay it somewhat, at least in the boys that were looked at in this study from Su et al. So the question we set out to look at was the fact that it's certainly been publicized that many things that had succeeded in the preclinical mouse model failed in patients. And people discussed why that was true. And there's certainly a discrepancy between the preclinical and clinical data. And suggested solutions have been using larger animal models that aren't difficult to breed, like some are. Animal models more representative of the human disease, and this is what we've tried to do with the rodents, and then less inter-animal variability, which the rodents don't have that much. They have some, but it's certainly much less than humans. And the skeletal muscles in our two rodent models have a very rapid progression of fibrosis and accumulation of TGF-beta, and that results in muscle regeneration really beginning to slow at 4 months of age, and then very limited thereafter. So that's much more like in humans, where there's not so much regeneration going on. And the standard MDX mouse has tremendous regenerative capacity. And so that's one of the reasons why we think it's so mild. So we think the D2-MDX is a better preclinical model of the skeletal pathology. So the issue then is, what about the cardiomyopathy? So it had been suggested that there was inherent hypertrophic cardiomyopathy in this DBA background that the D2-MDX is based on. But then we did a study that showed that that's really not true. There are some mutations in this line compared to some other lines, but they're benign in terms of disease causing. And so then we went on to characterize these mice, and we see that both the mice and the rats have a cardiac progression similar to what I just showed you for some of the human DMD patients. So we're looking at both histology and EKGs and doing echoes on the mice, very high resolution echoes. And what we see coming out of that is that, as I said, the background itself doesn't have a disease, and so they look pretty normal. But once you put that in the MDX background, now you start to see a rapid progression in the cardiac fibrosis. You see abnormal EKGs. You see a prolongation of what is called the QT interval, which basically means the ventricle's not repolarizing fast enough. It's very slow to repolarize, so it's got an extended calcium transient, and that's why it's not relaxing. But you can also see a number of other, besides this impaired repolarization of the left ventricle, you can see a number of measures that reflect diastolic dysfunction, which is progressive in the mice, up to the point where eventually, like in those humans I showed you, you leave this period of diastolic dysfunction where now the end diastolic volume is much smaller than it should be, and then you progress to a dilated cardiomyopathy. And that's what the mice do. They start off going through this phase, and then as you get cardiomyocyte death, you progress to this dilated cardiomyopathy and heart failure. But early on, you can detect this end diastolic volume decrease, which is stable for the first year before you start to then slowly progress toward the terminal phase of the cardiac disease. So the line itself doesn't have an inherent cardiomyopathy, as was reported by a couple of papers. And so it is an appropriate background strain for studying cardiomyopathy causing mutations. And it does recapitulate many features of what has been shown to occur in humans, and does progress to DCM and heart failure. So we've now been using these to re-examine molecules. So we looked at a couple of dozen small molecule therapeutics that had previously been shown to bring benefit in the C57 background, and most of them failed to show any benefit in the D2-MDX background. And especially in the heart, very few of them had any benefit for the heart. We've also been looking at the microdystrophins in clinical trials, and what we see with the microdystrophins that are in clinical trial is it does recapitulate largely what we're seeing in patients, in that it does have benefit to the skeletal muscles, but the benefit is not that it arrests the disease, it slows the disease progression, but the disease still progresses, both in the diaphragm, the heart, and in the limb muscles. So so they're not perfect. And so because of that, we've been trying to develop microdystrophins with other elements in them, other dystrophin elements that might do a better job of rescuing the heart and diaphragm. And at this point, we've got at least two that we've designed that seem to provide much more cardiac benefit than the constructs that are in clinical trials, although they haven't provided that much diaphragm benefit. So we don't know what it is that the diaphragm needs from dystrophin, and we don't know if it'll even fit in the microdystrophin sort of package. But we'll find that out hopefully over the next year. And so with that, I will stop and take any questions whenever we get to the question period.
Our next speaker is going to be Erik Henricson, talk about the CINRG Natural History Study and where that project is headed, which PPMD does have a long history with, and happy to continue supporting and working together.
Yeah, thanks, Erik. Everybody hear me all right? So I want to talk just for a little bit about the natural history studies that we've been doing in the CINRG group since the early 2000s, really. And a number of folks who are here at the conference had who are adults now were young children in the study. So we have a bunch of folks here who have actually contributed data as well. But just a brief history. So we started off in 2004 to 2016. We collected data on 440 families worldwide. And that was not just guys with DMD, but also data from parents and caregivers and family members in 9 countries and 24 different hospitals and clinics worldwide. We had over 3,500 patient visits, and that spanned, to use a public health term, over 1,600 person years. So a person, if you follow a person for a year, that's person year. So it's a lot of data. We're not the only folks in the world doing this. There's some excellent other projects. The Imaging DMD has some great natural history data, as well as the Italians and the UK group, who have all provided information that we can now start to kind of compare together to look at how worldwide populations with DMD look against our current care standards for folks with drug therapies and PT and things like that. So we started to look. We took the kitchen sink approach to this study, where we wanted to look at not only medical evaluations and strength and function evaluations, but also things that had to do with medical care utilization and medication use, and also looking at activity and participation assessment, quality of life, and sort of the lived experience of being a family with DMD, with the idea that we wanted to really describe not just strength and function on the sort of tissue or organ level, but also start to get an idea of the relationships between strength and function and activity and participation, and how that led to individual wellness and health overall. So this is kind of a little, this is the hard part about doing public health studies, is you have this big gap. And for us, it was almost 5 years where you start a project, everybody's killing themselves to get data. Families are coming in, they're doing tons of evaluations, everybody's working really hard, and no publications. Like, it seems like nothing's happening. And that's really tough, because it takes a long time to amass enough data from a study like this to actually start to do useful things with it. So from 2010 up until present day, we now have close to 60 different papers based on our data. And some of that is biomarkers and genotype-phenotype studies. Some of that is natural history and disease progression modeling information that is being used to develop clinical trials now. Some of that is actually data that's being used as comparison cohorts against the existing clinical trials that are out there right now, or ones that have now progressed to the point where we have drugs on the market. But also specifically things that development of clinical outcome measures as clinical trial tools, pulmonary health and function, cardiac health and function, and even a couple of forays into neurodevelopment and quality of life. So today, we ceased our data collection in 2016. And part of that was due to a lack of funding and the lack of funding mechanisms to get that done. PPMD actually helped us with some bridge funding for a final cohort that we had, and then to help move into some data analysis with a couple of sponsors. But in the interim, we've been working with a group of pharma sponsors, of whom Sarepta is the first one to help get us some funding to restart the study with a new cohort looking at contemporary measures, contemporary health, and what clinical care looks like today. And so that's kind of where we're starting. To just look back at impact, because I think it's important to revisit, we have, with the original study now, over 1,100 citations. Majority of those are basic research articles that where they've pulled some information, but we have 93 clinical articles that have cited this as well, including the ATS guidelines for pulmonary management, the American Heart Association cardiac management guidelines, the steroid practice guidelines from AAN, and the CDC care considerations. We most recently had the one that I found most recently was actually a guideline for DMD care by the government of Brazil. So it's pretty wide ranging, and the other groups who are doing similar kinds of studies are also seeing data used in that fashion. So this is my NASCAR slide. I always thought I should have one of those jackets with all the patches all over it. But this goes to show that this is definitely a collaborative team science group effort. This is industry, this is academia, this is advocacy groups, and it's everybody working together. And in the case of the initial study, when this kind of world of public health research in Duchenne muscular dystrophy was just evolving, also represented a whole lot of volunteer time from a whole lot of people. So today, with the help of Sarepta, and and our model is, the idea is that we can involve industry sponsors in a non-competitive space where natural history information is important for us in academia. It's important for industry sponsors who are working in parallel lines but all need the same kind of data. It's important for regulators. And so Sarepta is our first sponsor for this. We have a 36-month window to get the study up and running with a cohort of 110 participants. We're going to take all ages, all treatments. You basically have to have a moderate to severe dystrophinopathy of that looks like a Duchenne phenotype. We are in the process of doing our site selection right now. We're going to be in multiple countries around the world. We haven't nailed it down completely, but we have initial funding to get 10 centers going. This is a collaboration between UC Davis, so Craig McDonald and myself, Heather Gordish-Dressman, our biostatistician at George Washington University, and Dr. Tina Duong at Stanford, and as well Binghamton University with Dr. Yetra Pathut, who is going to head up our biomarkers program. The protocol itself is the assessment schedule is going to look pretty much like what we had before, where we're going to do quarterly evaluations for the first year, evaluations every 6 months in the second year, and then annually thereafter. And this is designed to be sort of a perpetual long-term follow-up study. So what this does is it gives us the ability to get some finely resolute measures of variability and reliability of measures sort of upfront for different cohorts, and then also look at getting information about long-term progression without burning everybody out. And so that is, we are in the process of getting this rolling. We hope to start with participant enrollment sometime starting in probably August or September, maybe October, depending on the site. And so we look forward to getting this out on the ground. This is my cast of thousands slide, and it just is a ton of people. And so again, hopefully we look forward to being able to provide key information to update care standards, develop new clinical trials, and look at real world evidence moving forward. Thanks very much, and I appreciate your time. Thanks, Erik.
Our next speaker is going to be Amber Sapp from Casimir. And some of you may have seen if you're in some of the other sessions, Casimir being, the DVA being used as an exploratory measure in some of those clinical trials.
Thanks, Erik. And thanks to PPMD for their ongoing support of our tool. They've really been instrumental for many years through the beginning inception to helping us with recruitment for our study. So we're really grateful for that. My name's Amber Sapp. I'm a physical therapist by training. I am now the neuromuscular solutions lead for Casimir and sort of guide and direct our Duchenne-based programs and some other neuromuscular programs we have. I also have an almost 17-year-old with Duchenne who is very sad that he's not here this week because this is one of his favorite events, but much to his disappointment, he is at his sister's dance competition. So I'll tell you guys a little background about the DVA, kind of where it came from, what it looks like from a patient caregiver perspective, where we are with our qualification pathway with FDA, and a little about our study called ARISE that we're running internally. So as you know, a lot of clinical trials have to determine what sort of outcome measures they're going to use on the onset of their study to determine, if they're seeing improvement, decline, or stability across the spectrum of disease. And a lot of these measures include time function tests, so things like the 4-minute stair climb or how far you can walk within 6 minutes. But as a parent, I think we see these things happening a little bit differently. We see the way that our kids compensate to maintain their independence. I remember the time that my son had gone from, eating with a fork like this to suddenly he was using, 2 hands to get fork to mouth. Or when he was younger, they're climbing the stairs step over step, and then suddenly they're doing this kind of step 2 pattern, or they're pulling themselves up with 2 hands on the handrails. These are all sort of compensations that we see the kids do over time, and they really represent a certain level of muscle weakness that's starting to happen. Some other tools that you see used as outcome measures use a 3-point scale. So we have the PUL, which is looking at upper extremity function, the North Star looking at more of an ambulatory participant. And they use these 3-point scales, right? So you've got, you score a 2 and you're uncompensated in your movement. A 1 represents some sort of compensation, and it's quite a large spectrum that it could, any sort of compensation. And then a 0 is that you can no longer perform the task. And so the Duchenne Video Assessment really wants to kind of fit into that 1 and understand where we are in that spectrum of compensation. So our Duchenne Video Assessment is a standardized way for caregivers to capture video recordings of their child at home in their more natural environment. And the tasks are pre-specified. And as this is really an overview, I'm not going to describe to you how we came up with this task list, but I'm happy to share some manuscripts if you guys are interested. Our concept of interest is ease of movement, not necessarily speed of movement. And the tasks that we've chosen represent meaningful tasks to patients and caregivers. One benefit is that it decreases this travel fatigue component, and kind of this medical anxiety that sometimes gets demonstrated in a clinic. My son was a part of several trials over the years, and when he was younger and stubborn, I remember, he would tell me, how'd you do on your run? And he would say, I didn't feel like doing it, and so he wouldn't do it. Or if he didn't like the provider that day, it's so-and-so, the PT, that he doesn't have good rapport with, he was stubborn and wouldn't perform it. And trying to translate that to kids is how, that data gets translated into a study, but we don't really account for that. And so what I like about our assessment is that it happens at home, and families are given a 2-week window to capture. So if your kid is stubborn one day and says, I'm not doing it, or if they don't feel well, you can choose another day within that window to capture. And in the end, we have certified physical therapists that go through a pretty rigorous certification program, and then they have to recertify on a yearly basis to continue to be raters for our studies. Now this is a task list that represents the entire composite of what our tool looks like. These are all activities of daily living or functional tasks. And again, these are identified by physical therapists and families and participants as meaningful to them. Now, not every participant will perform all of these tasks within an assessment. We branch our participants into functional cohorts, ranging from early ambulatory to late non-ambulatory, and based on which cohort you're divided into, you'll get a task list that's associated with that. So from a caregiver patient perspective, this is what it kind of looks like on your end. If you're a part of one of our studies, you download a mobile application. That's where your videos get taken within that app. You also receive a study kit, and that, again, is depending on which cohort your child is in. And it's going to include things like a tape measure and painter's tape. It helps us to standardize, the distance from the camera to the child, and it helps us to make sure that if we've asked you to walk 20 feet, you're indeed walking that distance. We also standardize everyone gets a set of plastic forks, and so everybody's using the same weight of fork to feed themselves on those tasks. And then caregivers and participants are asked to watch training videos that show them exactly how to capture the assessment. And they also get a training manual. And so I really like that way that we've approached that, because it sort of covers all different learning styles. If reading the manual is the way you learn best versus watching a video, it just really encompasses several learning styles. Like I said, you have a 2-week window to capture, and then we have data monitors on our end who will, as the videos are submitted, they take a look at those videos and make sure they meet our quality standards. And it's a pretty lengthy list of quality parameters, things like lighting, distance from the camera, making sure the clothes are really similar. They're not trying to do the activities in bulky long sleeves or a sweatshirt. And so if these videos don't meet our quality standards, then they may get sent back for a reshoot. Our reshoot rate currently is less than 10%. Some of the things that also get sent back for reshoot is if you capture a baseline video and your setup is in the office chair, and then your next video you're sitting on the couch for that task, that's going to get sent back, because we want to always compare apples to apples here. So this is an example from one of our internal studies. The task is standing up from the floor. So we just have asked the boys with a standardized script that they'll find within the app, stand up from the floor. And so some of the things you immediately notice is that he's compensating in some ways, right? He's got this kind of modified Gower maneuver he's doing. If we score him on a North Star assessment, he's a 1, right? He's compensated in some way. But what I really like is the way that we can kind of dig more granularly in here. So we can look that he does have this kind of Gower where he's on all fours, but his base of support is just slightly wider than shoulder width. He's not walking his hands along the floor to kind of help lift his trunk up. He's also not using his hands on his thighs. And he's not really straining to lift the trunk. Now, when physical therapists score these, they are unblurred so that we can see, this strain that appears on their face. It's a little easier to detect. So if we count up these compensations, you can see that he has 2 out of 8 for this scorecard, and he would be considered 25% severe, whereas 100% severity would mean that they can no longer perform the task. All right, so this video is the same kiddo. It's taken 30 weeks later. And I think that it's pretty obvious that he's got some decline. And so the way that we can quantify that decline, instead of this just being anecdotal, oh, he looks worse at this time point, on the scorecard, his base of support is now significantly wider than it was at the first time point. He's now having to walk both hands along the floor to lift his trunk, and he's using one hand to push off of his thigh. So his severity score now is 62.5%, but on a North Star, he's still a 1. And I think it's really important to note that, within 30 weeks, we see this happen, whereas a lot of times these measures take a full year, and they'll describe to you it's a year before you can detect change, and, our boys don't have a year. And I think our ultimate goal for parents and sponsors would be to have a shorter placebo-controlled study. I mean, we don't want a year to see movement on these activities. So I mentioned we're in this pathway with the FDA. It's called the qualification pathway. And we are currently collecting the remaining data that they've asked of us in order to become a qualified assessment in Duchenne. And we would be the only qualified assessment in Duchenne once this process is sort of complete. And what that really means is it's kind of the FDA's stamp of approval to say that your tool is well-defined and reliable to measure the concept of interest that you're describing. So we have an internal study called ARISE that will allow us to capture this last piece of longitudinal data to submit with our FDA qualification package. We're on target to have complete enrollment by October, and this we are enrolling participants 2 and up. We're also including a female group just as exploratory to see, are the movement patterns that we see in males with Duchenne similar to those that we see in females. And participants will be asked to collect a set of 6 videos over 2 years, and they're well compensated for their time. So currently, this is a little outdated. We're about 3/4 of the way to enrollment, which is really exciting. And we plan to submit our package to FDA at the end of summer. So if you guys would like more information about the study, you can scan the QR code. There's an email address, phone number, and one of our study coordinators will reach out to you. This is just a thank you slide for everyone who's contributed thus far to our tool. So thank you.
And our last speaker for the session is going to be John Soslow from Vanderbilt. And this research is part of one of PPMD's larger research initiatives, which is our cardiac initiative. And so we've had a lot of support in that area, and this work is helping to build that out.
Great. Thank you, Erik, for the introduction. Thank you for inviting me to speak. I also want to point out I'm here on behalf of multiple investigators, myself, Larry Markham, Chris Spurney, and then Chris Heyer, who's helping us with the microRNA. So I have no disclosures relevant to this talk. We're going to start with a little bit of background and talk about the DMD Cardiac Care Consortium, go through our proposal details and some of our preliminary data. And I'm just going to skip ahead because I think everybody here understands Duchenne muscular dystrophy and the issues around the skeletal muscle disease, but the cardiomyopathy is really our focus. And there's been data for many years that LV dysfunction decreases survival. And so we've known this for a while. And we also know that over the last 30, 40 years, survival has improved significantly. This is a study looking at boys born in different decades, and that you can see that the median survival has improved significantly, such that in the most recent era, it's around the 30s. However, with this improved survival, we do see much more of the cardiovascular disease. So it's really unmasked that cardiovascular phenotype. And cardiovascular disease is now the number one cause of mortality. So how do we evaluate cardiac disease? Well, clinically, as many of you know, symptoms are not great in Duchenne muscular dystrophy because of the skeletal muscle weakness. Electrocardiography can be helpful. We use it for a lot of different things, but it's relatively nonspecific. And then standard cardiac blood biomarkers are only really elevated later in disease. So it really leaves us with cardiac imaging as the gold standard, echo and cardiac MRI. They both have their strengths and weaknesses. Unfortunately, they both need to be done at a clinical center. So I can't easily get an echo or an MRI at an outside facility, and that can be an issue, especially during things like a global pandemic when my patients can't come to see me. So we really need novel, non-imaging based assessments to help with our evaluation clinically. In terms of therapy, we use standard adult and pediatric heart failure drugs. And we know that earlier therapy really improves the response. But the majority of studies are in a small number of patients, and we haven't really done standardization of the baseline medication regimens. And so most of us in the field really feel like we need novel disease-specific therapies to improve cardiovascular outcomes. What are the gaps to developing these? Well, there are no established clinical outcome measures or biomarkers in DMD cardiomyopathy. There's no agreement on endpoints for cardiovascular trials in DMD. We use LVEF and strain and some other things for the studies that have happened in the past, but the FDA has never really officially approved any of those surrogate outcome measures. DMD cardiovascular disease is also very variable, and so we need a lot more patients in order to do a successful study and demonstrate a change. And then we know that medications need to be started early for the best efficacy, but that is really too long a timeline for us. So if I start at 10 or 12 and I know I have to wait 10 or 15 years to see the mortality events that the FDA really wants, that's not going to work. It's too long a study, it's too expensive a study, and honestly, we want to get drugs to patients, and so we don't want a 10 or 15 year gap until patients get treated. So we really need surrogate outcome measures in order to evaluate these novel therapies. So that's pretty much the goal of the DMD Cardiovascular Care Consortium. We want to identify serum and imaging biomarkers that can be used as surrogate endpoints in clinical trials. We want to identify serum and imaging biomarkers or genetic variants that can predict progression so that we can risk stratify patients and decrease the number of patients that need to be enrolled. And we want to leverage the DMD CCC to begin clinical trials of novel therapeutics. So this is our current consortium. We started with 4 centers, and we've now expanded out to 8 over the years. And each of them has an interest and special expertise in cardiac care in Duchenne. And we've been productive. We feel like we've done a good job, and we have now 19 manuscripts, 20 abstracts, 2 grants funded, and multiple grants submitted and in preparation. And this is our natural history study that's FDA funded. So, and this is the study that we have sort of been able to use some of PPMD's money to add on to and really do some of our biomarker work. And so what we set up is a natural history study really focused on cardiomyopathy and doing very detailed cardiac MRI at baseline, 1 year and 2 years, as well as accelerometry, quantitative muscle testing, blood samples, and then trying to collect all the other things that we're all interested in as a group, like pulmonary function testing, et cetera. And so we have these standard operating procedures built out, and we've added sites slowly to get to a point where we feel really comfortable with each site and what they're able to provide in terms of the CMR and the actigraphy and everything else. And we now have over 150 patients enrolled in this study. So, back to the blood biomarker question. As I said, there's really a lot of advantages with being able to draw blood and get an assessment of where someone is. It allows us to do a remote assessment. It allows us to assess patients who might have poor echo windows, which is quite common in DMD, or who might not be able to go into a scanner because of contractures. And so there are a lot of really nice clinical advantages. We also could use this for the studies that we're interested in. So if it predicts progression of disease, it could allow us to have earlier therapy, but it could also be used as a surrogate outcome measure and help us risk stratify patients before starting a trial. So 2 of the substances, or 2 of the sort of groups of biomarkers we've been interested in are MMPs and microRNAs. And matrix metalloproteinases and tissue inhibitors of metalloproteinases that are substances that regulate extracellular matrix remodeling. They also regulate inflammation and apoptosis, or programmed cell death. And so all of those pathways are pretty critical in the progression of DMD cardiomyopathy. We're also interested in microRNAs, which are non-coding RNAs that play integral roles in mRNA regulation, gene expression, and cell-cell communication, again, all of which play significant roles in almost any disease process. And microRNAs also have the added potential with therapeutic implications, so that people think that we can start to give these microRNAs to help with disease. So our hypothesis was that we could evaluate microRNAs and MMPs and TIMPs to look at myocardial dysfunction and fibrosis in our DMD population. I'm going to go through some of our preliminary data. So we looked at MMPs, multiple MMPs and TIMPs in 42 DMD patients and 15 male controls. We showed that MMPs 1, 7, and 10 were increased in DMD and that TIMP4 was decreased. And when we compared the MMP levels to our cardiac MRIs, we found that MMP7 in particular correlated inversely with left ventricular ejection fraction and correlated directly with LV circumferential strain, so that as MMP7 increased, there was a worsening in function for both measures. We also categorized everybody as control, DMD without cardiomyopathy, and DMD with cardiomyopathy on the left panel, and showed that there was a tonal increase in MMP7 as you went from one category to another, and then looked at MMP7 and delayed enhancement, so SCAR in the heart, and showed again that controls were lower than DMD patients without delayed enhancement, which were lower than DMD patients with delayed enhancement. So moving on to our microRNA data, we looked at 26 DMD subjects, 13 with delayed enhancement or SCAR and 13 without, and compared them to 10 controls. This was a large 750 microRNA panel to do a kind of a pilot study to look for the microRNAs that we were most interested in. And we wanted to focus on microRNAs that had already been described in cardiovascular disease and were related to scar formation. And so these are 5 of those microRNAs that we found interesting. This on the left is MYR142.3p, and you can see that the healthy controls in the DMD patients without delayed enhancement, there on the left, have relatively similar numbers for MYR142.3p, whereas those with delayed enhancement have decreased MYR142.3p. And then the other 4 all showed a similar pattern where the DMD patients without delayed enhancement had much higher levels of, in this case, MYR30E and MYR375 than those with delayed enhancement, which implies a potential for a therapeutic benefit, although obviously much more needs to be done. And then again, the same pattern with MER206 and LET7C. So in conclusion, microRNA and MMPs, particularly MMP7, have potential as biomarkers in Duchenne. We are hopeful that they could be biomarkers of cardiac disease and or surrogate outcome measures. And then there's also some potential for microRNA therapeutic benefits. And our next steps, we have already sent 150 samples from our baseline study to Children's National. They are in the process of doing the microRNA analysis right now. And we are doing the MMP and TIMP analysis at Vanderbilt. And then we also plan, because we have this longitudinal data, to look at how the baseline values of microRNA and MMPs associate with longitudinal progression in DMD. And that's everything. I just want to thank everybody in our DMD Cardiac Care Consortium and everybody at Vanderbilt involved with the work. So thank you.
I'm actually going to have all of our panelists come up here. So we have a few minutes for questions. So if there's any questions from the audience, raise your hand. Lauren can run a mic over to you. And these, yeah. While we're getting started, there is, I think, one that's always helpful. We've done some polling before with the community in terms of participating in natural history studies. One of the concerns always being, can I be on approved therapies? Can I be on an investigational therapy and still participate? So for the 3 natural history studies, is that something that patients are able to be on an investigational therapy or an approved therapy and still participate?
Sure. Actually, that's one of the things that we're quite interested in, is looking at kind of what contemporary care looks like and what the impact of that is on the progression of the disease. You've probably seen a number of slides, here in both presentations today and previously, looking at how, survival or onset of certain, loss of milestones has shifted over the years. We're looking to add new lines to that, basically. And so it's important for us to be able to have a population who's balanced, that represents what's going on in clinics now.
Same with us. I will say the caveat, though, you're allowed to be on any investigational therapy and you're allowed to change any medicines you want to change, unless we think it's an investigational therapy specifically for cardiovascular disease, just because we want to see what the natural progression is with current sort of standard of care. So any changes can be made. You can be on other studies. So we have limited gene therapy, and we've limited things like the Affitrabant study, which actually we're also a part of. And so those are kind of separated. But anything else, you're welcome to be on. And we completely are open to joining. And then if you have another study and we think there's a cardiac effect, we're not going to try to keep anybody in. We want everybody to get the treatment that they want and their children need. So.
Yeah, and likewise with our study, for our ARISE study, we are capturing, through questionnaire data, classification of drugs that participants are on, but we're not limiting that to one another. So.
Great, thank you. Lee, you talked a little bit, obviously, the development of these new models, and that's something we want to see advancements in terms of development and preclinical, so we have better prediction of what will translate into the clinic, and again, fail faster if we are going to fail, and get to answers faster. Are you seeing adoption of kind of new models as they're being developed by companies? I know you work with a lot of companies, or are we still relying on MDX?
Well, we're doing assessments for a number of companies. So they've come to us and asked us to assess, mostly in the mice. Some of them have asked us to also use the rats. So I don't know that they want to set up colonies as much as they would rather come to people that have established colonies.
Hi, this question is for Dr. Lumley. So one of the gene therapy problems we see is that due to the window of the microdystrophin, we have certain exons excluded and certain ones we don't want to test and whatnot. Do you see, since yours is a full-length dystrophin, do you see any sort of problems with the exons, ones that wouldn't be involved in any of your studies or wouldn't be available to or anything like that?
The simple answer is that we don't know. I think we're all trying to get to the bottom of the immune effects that we're seeing with the microdystrophin. So I think that remains a risk for any gene therapy in development. So we don't know, and that's something that we're going to have to consider very, very carefully in clinical trial design. Very speculative, but there is an argument that if the microdystrophins, the truncated dystrophins are foreign proteins, and that might be the crux of it, is exposure to an abnormal epitope that the body doesn't see because of abnormal folding. So potentially, or theoretically, if you deliver a full-length protein that undergoes normal conformational, post-translational modifications and protein binding, you might not see that. But that's 100% speculative. And, when we get to clinic, we need to be really thoughtful about how we design it. We want to be inclusive, and you want to include all patients in clinical trials, but you also want to maximize the probability of success, and that will be one of the critical questions that when we get there, we'll take advice from, clinician experts about inclusion, exclusion criteria.
All right. Well, I'd like to thank all of our panelists for those great presentations today and everyone for joining us. So we're going to break for this session. There will be lunch available in the main room where we had the gathering last night. So thank you, everyone, and thank you again to our speakers.